welcome to the fourth day of the IChemE Student Summit yesterday, today, and tomorrow. I'd like to start by acknowledging the traditional owners of country throughout Australia and recognize their connection to the land, waters, and culture. We pay our respects to their elders, past, present, and emerging. In this session, we'll be focusing on the world of pharmaceuticals and biotechnology. This is a very lively world and something that I think is on the forefront of all of our minds, especially under the shadow of a pandemic. It's an incredibly diverse world, and our 5 speakers today will be showcasing this. We'll be joined by Professor Volker Hessel from the University of Adelaide, from Dr. Celine Valery from RMIT University, Dr. Pau Loke from the University of Nottingham in Malaysia, from Russell McGifford from the CANN group, and from Professor Charles Cooney at the Massachusetts Institute of Technology. So to start off, I'll be introducing Professor Volker Hessel. He is the Deputy Dean in Research and Professor at the School of Chemical Engineering and Advanced Materials at the University of Adelaide. He is the Research Director of Adelaide Sustainable Planetary and Space Resources, and has been the author of or co-author of over 470 peer-reviewed papers. Thank you for joining us, Professor Hessel. The floor will be yours.
Yeah, hello and good afternoon to everyone. And Nimish, thanks for the kind introduction. Yes, I want to convince you that part of future pharmaceutical science and engineering will happen some kilometers above us in the outer space on platforms such as ISS, and that we also will start to manufacture pharmaceuticals on orbit, on planets, and maybe also with benefit for Earth. Now, we follow largely what the big space agency tell us to do. And naturally, we orient on NASA here. They made this document in the upper right side. And that is a document maybe 100 pages long, and it tells all details. And okay, essentially, we have to build a new world. And if you build a new world, you have to do quite a lot. And this is given in this spider net diagram, this radar plot. And I will start with metal flow extraction, but then I will come to the moon dust tablet, that you find on the left side, the LUNAR tablet. And NASA doesn't make our job easy. There's a new document, the 2020 technology taxonomy. So that has to be considered in the next steps then. Now, as you all know, we have COVID, and COVID is changing the manufacturing in the world. It's actually accelerating what anyhow will happen. We will go 200 years back. So we will manufacture in smaller units, self-sufficient revenue district. And experts call that fractal economy. So we move a bit away from the global economy, at least that is what people think here in Australia and somewhere else, but with the most modern technology, naturally, not with 200 years old technology. So we come from what is called centralized, large-scale, long lead time, forecast-driven production to decentralized, autonomous, near end-user manufacturing. And what could be a better test case for such new technologies than the space? On the moon, we are helpless, we are alone. We have to produce very autonomous. Now, in the following slides, I want to show you need technology disruption. You cannot just take the same technology from Earth to moon. It's too different. You have to be very circular, make no waste, and you have to process in habitats. I call this sustainability here, but better should have called it process integration habitats. So just remember disruptions, circular economy, and habitats, and I will give evidence for that. Now we all know there are some asteroids around, some closer, some more distant, and they are basically like Fort Knox. They contain a lot of metals, and also the very expensive one like platinum, but even the normal ones like cobalt makes the asteroids enormous rich. So each asteroid, and as small as it is many, many times the whole money which USA, for example, spends every year. So it makes sense to look at that despite the technical difficulties. Now, what you have to do there is you have to work circular and with minimal use of resources. What I want to show here is what we process and what we think is an asteroid solution for minerals processing. It's basically flowing metal. It's 70% metal per weight, roundabout, and it's 30% water. And if you cool it a little bit down, then you get this big stone over there. So that's not the normal fluid we are on Earth, and I would claim that fluids have never been tested, at least not economically, and that is what we are doing now. And we're doing it with real asteroids. We have this meteoroid Campo di Celio. Now, we're doing it in a Corning microreactor, and such reactors are many hundred times make pharmaceutical production in the world, mainly in China. And if you want to see what that is, I can show you. That is the whole reactor here, which you see. So you get an idea that it is not really small. And if you use it for pharmaceutical production, it's as big as a room. Now, what we see there, without going into details, are flow patterns which are not typical for Earth fluids. That are space fluids flow pattern, different flow patterns, and they have different engineering quantities. Reynolds number, D number, if you know what that is. So we have to reinvent, in a way, chemical engineering when we go to space. Now, with the first steps done here in Adelaide, we can show that for the lower concentrated solution, that is the left side, we can have a perfect extraction as we know it. We are selective between cobalt and nickel. And if we go higher in concentration, so to the space fluids, that's what you see on the right side, you see there's a drop. We still got in the first round a reasonable extraction, and we can do extractions multiple times. So that is not a roadblocker, what we see over there, but we have to make new learnings with those space fluids. Now, to show a circular economy, this plot is here. Main message is we have to combine in space and also on Earth in future unusual areas, scientists which usually do not talk to each other. Here, space mining and space farming. Energy is a third topic which will come into play. On the left side, you see creeps, that is lunar phosphate, these stones there, and they go into a mining machine as I've just shown to you. And we think nature is doing best to accumulate that phosphate. So we accumulate lettuce. Lettuce is famous for being very phosphate rich. So we eat the lettuce in a space farm, which I will show in a second, and we take the roots for making phosphate fertilizer in the furnace. And you see on the right side that this works quite nicely. And instead of using aggressive hydrochloric acid, as it's used in chemical industry today, we use oxalic acid from sourshops, which basically does the same job. So this is a green and circular process. Now, as I already mentioned, we are also busy with space farming with our colleagues from Wade. We have the very large agricultural campus here in Adelaide. And we have this machine from Airbus, the so-called random positioning machine. And that is the golden standard to make microgravity on Earth, at least if you want to make it a little bit longer, not in a falling tower. And we need to make microgravity for 1 week or longer to have results with plants. And we are underway to make green plant studies under microgravity with this machine. And you see also the drawing of our space greenhouse. I could have also given a photo, it's too new, so we have manufactured it, believe me, and we are investigating now such space greenhouses. Such things are not done by one person. We have here a few what is called requirements engineering that is famous for making computers and also in aerospace. And from requirements engineering, you know that you have to bring together very different specialists. Look in the upper left side, we have chemical engineers. Okay, I talk to chemical engineers and pharmaceutical ones, but we also need crop physiologists. We need process sustainability engineers, hydrology scientists. Long story short means we have to bring people together which never talked before together, and that is a new challenge. So team working is ultimate in space to make good research. Now I come to the pharmaceutical engineering. Astronauts get sick. They're just humans. And this happens from the first moment of space exploration. Here's Apollo 7 given where one astronaut was sick. And this was just 1 or 1.5 years before the famous moon landing by Neil Armstrong. Now, there are statistics about this. And when you see as a table, you see their experience with submarines. And we Australians have experience with the Antarctic mission. Their experience with the space shuttle and experience with ISS. Long story short means the longer you stay in space, the more extreme it gets. So as you see, the ISS cases are much higher. The medicines do not really work in space as good as they work on Earth. So that makes the situation more complicated. And we see below some typical diseases which happen. And you see 25 to 75% of all crew members get sick. So this is serious. And what comes on top is the cosmic ray decompose the medicines. And that is where our invention sets in. So we want to make a moon dust tablet, as we call it. It has two features. It should be cosmic ray stable, much more than normal tablets are. And second, it should be made only by materials from moon. And I think you all know that half of the excipients are organic materials and like waxes and so on, and we cannot use them. They do not grow on moon in a way, so we have to use a moon material. And we will have a launch in September with Space Tango, and I'm very committed to that company who made that possible. Now, all those materials exist in one way or the other on moon. This is shown the tablet here. The moon has lower lands and higher lands, and the distribution is different. Where Neil Armstrong landed, this is full of titanium dioxide, this Mare Tranquillium. So we know quite a lot about moon, and our knowledge gets more and more. This Clementine mission, that is a satellite, an analytical satellite with spectrometers, gave us good ideas where to find the resources on moon. So we know where to put the mines in moon in future. Now, there's also good news. There's a lot of variety of materials in the solar system. And we have also materials which are not known on Earth, or at least not in this shape. So silica exists in many, many polymorphs. It's given here, and they have different crystal systems. And a few of them are not known in Earth or rarely known. So we have new materials, so we could make new types of excipient. So that gives some chances and opportunities. Now, what we made is the moon dust tablet, and you can see the tablet over here. That are the red tablets. They are red because they are made with iron oxide. And you see that is exactly how commercial ibuprofen is made. Not all of it, but some of it has this red outer shell. And that is just the colorant. But we use that colorant as cosmic ray shield. That is at least the idea. We will see if that works. Now, we send those tablets to ISS, and we send them in blisters. That is what you see here. This contains 20 tablets, the aluminum blister. And we make also smaller blisters, bigger and smaller. And I will explain you in a second why we do it. So they contain a number of tablets, and that will be tested on ISS in around about 3 months. Now, what are the components of our tablets? We have chosen ibuprofen because ibuprofen is probably one of the best studied medicines according for UV decomposition on this planet. The numerous studies on UV decomposition, that is not the only dangerous cosmic ray. We have high energy protons. There are naturally no or hardly any studies of ibuprofen on this, but that is why we go to ISS. We have combined it with vitamin C. Quite common that medicines are combined with vitamins. We have even very modern encapsulated flavors, but that is not the important thing. Finally, the important thing is the excipients. We have chosen them carefully, and we have used Earth materials which come as close to moon materials as possible. And the good news is, I was just yesterday told we get real moon material and can work with that in future. So we made 60 tablets of it in ISS. It's a passive payload experiment, as the experts call it. And the very special thing is we have 6 tablets outside ISS. So in the true outer space, cold, vacuum, all those quantities. What we can make within the limits of our experiments, we can make a variation of the supposed cosmic ray sensitive compounds that are the organic ones, ibuprofen, vitamin C, and the flavors, and we will analyze them when they come back. We can make a variation of the excipient mix as a whole. I would have loved to vary all excipients, but then we would have to make 600 tablets, and that was not possible for cost reasons. We vary the supposed key excipients for cosmic ray stability. This is the titanium dioxide and the iron oxide. And we have a small check on repeatability because if it's a single experiment, you never know if that is significant. But naturally, we cannot make 10 repetitive tests. So we had to make a compromise with space research. Now, Where is this happens? It happens in the Dignity module at ISS. Above, you see a photo in 2002 when this module was empty, but now you see there's quite some life going on. The astronauts working over there in this 20 years old module at ISS. So there the tablets will be stored, and they are stored in what Space Tango calls a cube lab. That is the outer frame, which you see on the upper left side, and this red and white subunits. This is what they call Tango Lab. And in one of those Tango labs, our tablets are packed. And they are stored for half a year. And in April, they will come down 100 kilometers in front of Virginia. And then they will be taken and shipped back to us. And hopefully, we will see something. Now, the outer space station, that is the so-called MISSE station, which you see above. And that is run by Alpha Space. And the cargo carrier, which brings our tablets back, that is from Northrop Grumman, that is the Cygnus capsule. Yeah. Then we work in parallel on a process design study with Aspen process modeling for making a moon factory. Now on Moon, we don't have really zero gravity, but we have a very low gravity. So what I sorted here out was which of the pharmaceutical formulation operations might work under zero gravity and which might not work in the way of a traffic light, yeah, red, yellow, and green. And I suppose some that the blue ones might even work better for reasons which I cannot now detail here. So we have a bit an idea what could basically work on Moon. Naturally, on ISS, we have a little bit better situation inside. And the last slide, let me see. It is, I have difficult to assess it. Yeah, now I can assess it. See, I hope the system will move to the last slide. Yeah. Okay. What you see here is we are, to our best knowledge, the first process experiment on ISS on that kind, which is led by an Australian researcher. So not just with a participation, and it's led in this case by me together with Space Tango. And we received quite some attention. A photo is here with our premier, Stephen Marshall. We also had Scott Morrison, the Prime Minister of Australia, at our booth. And you see internationally, I could show you 60 or 100 web releases up to Argentina, where it's translated in Spanish. Now, the last slide, and now I'm at the end, and I think I was not longer than I expected to be. I like to end, I'm German, with a great German philosopher, which is Immanuel Kant, and he said, two things awe me most, the starry sky above me and the moral law within me. I think that is a good quote, and I'm very passionate about space research, and I hope I could bring that a bit over to you. Thank you so much.
Well, thank you very much for your presentation, Professor Hessel. It's certainly a very unique one. For the audience, Professor Hessel will have to leave now, but for all our other presenters, you may ask questions in the Q&A chat, and if we have the time, we will answer them. All right, thank you. Now handing it off to Dr. Celine Valery. She's the leader of the Peptide Nanotechnology Laboratory and a senior lecturer at RMIT University in Melbourne, and has a background in chemical engineering, physical chemistry, and pharmacy. She's contributed to the European and FDA approval of the first ever hydrogel based on therapeutic peptide formulations. Thank you, Dr. Valery.
Yeah. Okay, great. Thank you very much. Well, first, thank you for the invitation. I think it's a great initiative, especially in those times, to try and gather a little bit all together and talk about inspiring science, or at least what we are passionate about. You are chemical engineering students. I did chemical engineering, and it opens really a very broad diversity of opportunities for you to work in very diverse fields with this background. So I'm really happy to talk after Professor Hessel, because he talked about space, and it was very, very inspiring, this lab in ISS and making drugs in microgravity and formulation. So very inspiring talk. And I'm actually going to talk about the other end of the spectrum. So we come back to Earth, but we're going to go into another universe, which is, I think, as fascinating, which is the universe at nanoscale. Right? That feels also sometimes being a little bit in space. And I'm going to talk about some nanomaterials, nanostructures that I work on, and which are made of biological molecules. And you'll see that we can do so much with that. Right? So first, just to get back on Earth and get back to the nanoscale, nano is very, very small. Okay. If you look at the bottom of that picture, what I'm going to talk about today is under 100 nanometers. Right, the characteristic size is under 100 nanometers. Probably in that figure, the closest structure would be the carbon nanotubes. Right, so you see that carbon nanotubes, where they characteristic dimensions, so the diameter is 100,000 times smaller, right, than a strand of hair. So that's what I'm going to talk about. I'm going to talk about that scale. So, nanotechnology in pharmaceutical products. Well, nanotechnology is very useful in pharmaceutics because we can do innovative drug delivery, we can encapsulate drug, we can change for instance, the solubility of a drug or the absorption of a drug, we can change the release of the drug from the vehicle. And so we can do plenty of things if we manage to fabricate at nano. Okay? The issue is that, well, we don't have machines that can fabricate structures at those dimensions. Okay? And we actually encounter very specific properties for the materials at those dimensions. So how are we going to make them? Well, we're going to use chemistry. And using the right chemistry, we're going to be able to basically build structures, just like Lego. We're going to play with nano-Legos. And if we want pharmaceutical applications, well, we'd better play with biocompatible molecules or building blocks. And so these are generally proteins, peptides. Peptides are mini proteins. Right, so they cost less, so that's why we're so interested in this, or lipids, or we can even combine peptides and lipids or protein and lipids. Right? So for instance, as a first quite theoretical example, but I'm going to show you non-theoretical, real examples that actually are on the market. You can see that that's an example of peptide molecules when they come together. So if you go on the left, they can form an aggregate that is not structured, so won't have, let's say, specific properties. But on the right on that figure, if we start having an anisotropic shape, so starting to build geometric shapes, we can start having specific properties, such as having a gel. And why do I like to play with peptides and other people like to play with peptides? Well, they're actually perfect building blocks to make those to make nano biomaterials. That's the way we call them. Right? You can consider peptides as nanobricks, okay? Mini molecules. They are just amino acids, sequences of amino acids, right? And they also have properties to self-assemble by hydrogen bonding. Okay, between the carbonyls. So I know I'm talking to chemists, so I can say a little bit of words of chemistry. And when you start playing with the building blocks and non-covalent interactions, well, you can actually start playing with shapes, and we make different nano shapes. All right, so one example that I'm actually working on at the moment, there are a few things I'm going to show you that I'm working on with my group and collaborators. So one big problem, a global threat, is antimicrobial resistance. Okay, it's not only having a new virus, it's can also be having bacteria that become resistant, and we can't treat them. Okay, we don't have any cure because they are resistant to all the antibiotics that we know. And so a lot of people are trying to make new antibiotic drugs. And peptides are actually quite promising candidates for that. One reason being that bacteria found many clever ways to build resistance against non-peptide drugs. But peptide drugs, if we design them well, they're going to destroy the membrane of the bacteria, and they don't have many ways to be resistant about that. This is a very simplistic view on this. I'm going to focus on the nano. So what we made some peptides, as I said, that can self-assemble. So self-assemble means we just put them in water, and it's going to form a hydrogel. We don't need to do anything. We just need to place them in water. That's going to form a gel. And it forms a gel because those peptides come together to form nanofibrils. And because we have these structures in water, they're going to retain the water and form a gel. Okay? Now, the interesting thing is that if we dilute this gel, the fibrils are going to slowly release the peptide. Okay, so if you make fibrils with an antimicrobial peptide, you can have a gel that will slowly release antimicrobials. Okay, we can make also many other things with that. Okay, other examples. So this is going into really solid nanomaterials. Well, we starting playing with making nanostructures that could be used eventually as filters or just to coat surfaces. And because of the very, very small dimensions, where we know that they can be antibacterial, and it's actually the way that butterflies' wings are made. They have nanospikes, and these nanospikes actually are going to act as antibacterial surfaces. Right? They're just going to pierce the bacterial membrane. So it's very morphological interaction. So of course, at the moment, with other materials and peptides, people manage to make those nanospikes. Well, those macrospikes, sorry. And we're trying to make nanospikes because we think if we go nano, we may have a chance to actually interact and trap viruses. Right? So you see that you can really play with those structures. So what do we use to actually study and know that we have those nanostructures? So we use a range of biophysical techniques that span from characterizing from the molecular scale to the macro scales. I won't go too much into details, but just showing you that we use a range of techniques that go from X-ray scattering, X-ray crystallography, infrared spectroscopy, Raman spectroscopy, atomic force microscopy, electron microscopy, right? So a broad range of techniques that you hear of in your chemical engineering studies. We actually use them. And in Australia and in Melbourne, where I'm from, where I'm working, I'm not, well, you can hear from my accent that I'm not from Melbourne. But, well, I am currently in lockdown. But when we are not in lockdown, we can actually go to the Australian synchrotron, which is in Clayton. And that's where we can do very powerful small angle or very powerful X-ray scattering, for instance. So an example of applications, because I told you we can do this, but I haven't shown you actually things that have been definitely done and on the market. That was the very first product I've worked on. So I spent a little bit of time working as a researcher in the pharmaceutical industry before joining academia. And I've been working on this hydrogel. So same principle as what I showed you for the antimicrobial, that we start from a drug that is a peptide and that forms gels. And these gels are nanotubes, and we can inject those gels under the skin, and they release the drug for over 1 month. Now, if we just inject the drug not as a gel, so as a much lower concentration, it's going to be attacked by enzymes, and the half-life of peptides is known to be around maximum 10 minutes. So by forming that gel, we actually have a depot formulation, so something under the skin that is going to last 1 month, right? Instead of 10 minutes. So that's quite advantageous. So this drug is on the market now. Okay. So of course, from that, I've been working on trying to design more molecules that make nanotubes. Okay. Something else that we can do, where we can use those structures to form or to template silica nanomaterials. So Professor Hessel just talked about silica, and there are some pharmaceutical formulations that are being studied with silica nanoparticles, for instance, on which you can graft many things. Where here we managed to find a way to make silica nanotubes, which are hollow in the middle, right? Just by using a peptide nanotubes, a silica precursor, and we can deposit the silica nanotube. So applications of this where we could think of nanoneedles, so injections that are harmless, for instance, or again, we could think of antimicrobial structures. Now, quite a surprising applications that I've been working on too with my collaborators is that we also used those nanofibers, well, in this case, it's nanoribbons that we managed to make with a very small peptide. And we worked with some collaborators that are interested in semi-conductor. Okay, so they modified the peptide chemically to have some highly electron conducting moieties, and they ended up with having nanoribbons that are supraconductors that you can insert in transistors. So this is also published. Now, well, I think I'm also very fast. We're very fast today. So some perspectives that we're thinking of and that we are making. So now in my group, we are interested in making metallic nanomaterials or composite nanomaterials, especially with gold and silver. Because there are a lot of health and pharmaceutical applications with metallic nanostructures. For instance, you may know that gold nanoparticles can be used to target cancer cells. So there are a lot of interesting properties for metallic nanoparticles. So of course, we're thinking of making metallic nanorods because there are also some interesting properties. And in the case of silver, for instance, where silver is antibacterial, so that might be another advantage to make nanosilver. And our latest thinking in the lab is to make silver nanotubes. I can show you at a normal scale because I actually have samples here. What they look like. Okay, so I don't know if you will see here. So here that's a very early stage, so it's a little bit red, but you actually have silver nanotubes in there. Of course, it's nano, you're not going to be able to see it. Okay, and here we have a higher concentration of them. So you see we have a deposit, and they are silver nanotubes. So basically, we're playing at the nanoscale, and these are pharmaceutical, a lot of pharmaceutical applications. So, in research, you never work alone. I think Professor Hessel insisted on that. It's always a team of people with different expertise. So it's not anymore the crazy scientist on his own in his lab. We are always a team and collaborating widely with people who are experts in different things. So here I have where the HDR students, my PhD students and MSc students that I've been working on and that are working on those topics, my collaborators at RMIT University, but we also have collaborators in New Zealand and in Switzerland. And well, some list of articles that we've published, but if you're interested, just look at my profile on Google Scholar, or you can send me an email if you're interested in the bio nano. So that's a little bit of bio nano that I tried to present today. So I don't know, we've probably have time for some questions. Nimish.
Hello.
Yeah.
Yeah, I don't think there are any audience questions at the moment. If any pop up, I will let you know. Thank you so much for your presentation. It's especially awesome to have someone from my university who has taught me several classes already. So thank you very much. It's always a pleasure to hear from you.
Thank you, Nimish, and thank you for organizing. That's great initiative.
No worries. All right.
Yeah.
I will now hand off the talk to Russell McGifford. He is a pharmaceutical manufacturing specialist at the CANN Group, which is the first company to be issued a cannabis research license by the Australian Government's Office of Drug Control. Russell, I will get your slides up in a second.
All right. Just had to sort out a bit of a mic issue there, but we're all good. So yeah, thank you, Nimish. I am with CANN Group at the moment, so medicinal cannabis. We were the first company in Australia to get a medicinal cannabis research license, also the first one to get a production license as well. So it's an exciting industry at the moment. It's a rapidly growing one. There's 29 licensed medicinal cannabis manufacturers currently in Australia. There's over 1,000 applications for licenses that are being processed. And on top of that, there's currently around 18,500 people in Australia who are being prescribed medicinal cannabis, and that number is growing substantially every month and every quarter. And there's a lot of interest worldwide in this. So Australia is definitely not the only country to have medicinal cannabis industries that are starting to grow. Canada is probably the world leader in that sense. And Europe in particular are picking up medicinal cannabis quite heavily, as are countries across the entire globe. And in fact, Australia's in a bit of a lucky position though, because we're approaching it from a very much a medicine-led sense. Some other countries are approaching it a bit more recreationally. And the net result is that, combined with kind of the pharmaceutical talent we have in Australia, means that we're becoming a bit of a world leader in the industry, and in particular, countries such as Germany and Holland are looking to us for product to import into their markets. All right, we'll start some slides when they show up. I can continue talking to white screen if need be. Do you know if the slides are on the way, Nimish, or am I having a technical issue? Yeah, that's right.
They
so make sure
should
it's not me.
be up now. I will make sure that they are.
All right, let me.
Okay. Because looks like there's scroll through my screen. Okay, all good.
Right, come in. Excellent. So the opening image I've got here is actually from a past life of mine. So immediately after uni, I worked in botanical extraction for not cannabis, but for poppies. And this is a picture from a field in Tasmania, which you might be interested to know. At one point in the recent future, Tasmania actually supplied something like 60% of the world's narcotic drugs, so morphine, codeine, thebaine, that kind of thing. So it's actually quite a big industry in Australia. And that's what I want to talk about today, botanical extraction. We've seen some amazing technology out of Professor Volker and also out of Professor Valery's groups around what can be done with high-tech manufacturing pharmaceuticals, but there's still quite a large segment of the pharmaceutical industry that's derived from plant substances. And in some cases, such as morphine or even nicotine, it's taken directly from the plant in the form it is in the plant. But it's also quite common that, for instance, with THC or CBD for medicinal cannabis, or when you look at things like codeine or oxycodone, there's slight chemical modifications made to that raw material before it's put into an actual drug. And then there's also the herbal and complementary medicine market, which is quite large, substantial employer in Australia and in fact worldwide. So botanical sources for drugs are definitely a big part of the pharmaceutical industry. And being a primary producer like Australia, they're a big part of the industry here as well. And it's not really a surprise. Traditionally, pharmaceuticals was all about plants. As chemistry students, I'm sure you've all done the first year chemistry experiment of turning silicic acid into salicylate. And that came from willow bark originally. So chewing willow bark was a good way to work for upset stomachs. There's a few other examples here. There's foxglove, native to Europe and a few other countries. That gives us digoxin, which is important heart medication. Ephedra is a Chinese herb. That gives us ephedrine, hence pseudoephedrine, all those cold and flu tablets. And then things like quinine from Kinchina. There's many, many examples of plants which gives us drug products. And in a lot of cases, harvesting and extracting the products from the plants themselves is still the most effective way to go. Before I get into the meat of the talk, just a quick word on regulation as well. This talk's meant to be very much around what it's like in the industry at the moment, because many of you may wind up in this industry. It's kind of a bit of an overview. And something I want to talk on is if you do wind up working in the pharmaceutical industry, or particularly in the extraction industries dealing with either cannabis or poppies, there's a lot of regulation out there, and it's important. But when you're dealing with pharmaceuticals, which need to be made to a very high standard for patient safety, and then you add on top of that the fact you're dealing with controlled substances that are actually illegal in most countries, there's a lot of regulatory overhead. And working carefully with the regulatory and quality departments of the company you're in, and then working carefully with local and international governments is a key part of negotiating this landscape. So I won't dive too much into this, but it's just something to keep your mind of. It's not something to be afraid of, but it's something to read up a bit on, particularly if you're going to be moving into these sectors. So what I do want to talk about today is just a couple of general points. First of all, I want to kind of give you an overview of what the standard approach to botanical extraction is. It's very blanket, it's very generic, but it covers particularly the industries that I'm used to, and it also covers a lot of other primary industries and near primary industries in Australia, such as essential oil production. And also kind of want to show you some of the key challenges around the accepted technology that's out there and the typical way things are done. And then following on from that, I want to show you just some of the upcoming technology, which has been around in some spaces for a while now, but is starting to become more and more adopted, and show you why that's important. So first up, if we're talking about medicinal extraction of drugs from plants, you need the plants. So the examples I've got here are all cannabis. And the reason I've gone for this option is that it kind of gives you an idea of the range of situations in which these plants can be grown. None of this is particularly shocking. It's all pretty intuitive. But I just want to highlight that while broadacre, which is you're growing an open field growth, like you can see on the left here, is a very common way to do it. And it's a very good way to get really high volumes of product. Particularly with higher value individual plants such as cannabis, there's a big drive to start moving towards either greenhouse growth, where you get a lot more control over around pests coming in as well as environmental factors, or potentially moving to full indoor growth, which in that situation you have complete control over everything to do with feeding, with pests, with environmental conditions, with lighting conditions, with photo period. You get really good control over how the plant's grown. And that's quite important to make sure that you're delivering a consistent quality product out of the plant at the end of the day. If you were to work in broadacre, it's very agricultural, unsurprisingly. It's very much local farming practices supported with some good agronomy. But as you move more into greenhouse and indoor growth, this starts becoming a lot more specific engineering challenges that need to be addressed. And it becomes an interesting little interplay between what the plants need and what the facility can actually deliver. So, not too exciting, but that's how you grow your plants. Next step is harvest. Once again, nothing earth-breaking here, but these are some of the major parts of this industry. And if you want it working, you'll be involved in all of them. Now, if you're going broadacre, chances are your harvest is pretty standard farming harvested. In a country like Australia, it's largely things like combined harvesters. And the product you get out of that tends to be a mix of everything that was in the field at the time, which is one of the drawbacks with that broadacre method, is you wind up with not only the plant itself, you'll wind up with the non-target parts of the plants, you'll wind up with seeds, you'll wind up with weeds, you'll wind up with insects. You kind of get everything out of the field. And then across the top there, you can see there's a harvester. The next image there is actually a pile of poppy straw that's been processed and is ready to be put into the extraction. In the background, you'll see there's a little yellow loader. Gives the idea of the scale of this. It's huge volumes of material. And the last picture there is roughly what you'd expect for about a single year's worth of poppy seed harvested from these processes. It gives you the idea of just the amount of material that's being brought in. It's a big industry. Each of those bags is probably around 1.5 tons of poppy seed. On the bottom, comparatively, we've got what it's like if you're more in a greenhouse or an indoor growth environment. Now, greenhouses aren't new technology by any stretch, but the use of them for medicinal plant production is relatively new, and particularly in terms of cannabis, the industry is still kind of figuring its way out through a lot of the challenges. So you'll find it's a lot more manual labor intensive. On the left, that's how you harvest the cannabis plants. You literally go in and cut them off from the growth media. And then there's a bunch of different ways to handle it past that. You might be physically cutting the flower off of the plant and trimming it down to make it appealing for the consumer. Or you might be using some semi-automated methods. On the right there, you have a trimmer, which is just a case of you feed in the flower and leaf material at one end, and it goes past a set of blades, and at the other end, you'll get out trimmed flower. It's really basic stuff. It does the job, but the truth is there's actually a lot of ground to be covered and a lot of potential for some enterprising entrepreneur to go in there and really figure out some of these engineering challenges to make the harvest more efficient. So you've gone through, you've grown new plants, you've harvested the plants and pulled out the parts that contain your real drug products. What's the next step? Now, if you're dealing with cannabis flower as the drug itself, and it does exist in some markets, such as Bedrocan out of Holland, you're kind of there, you've got your drug. But in the majority of cases, you need to pull the target material out and come out with the raw drug material from that. And the simplest way to do it is you simply take the plant material and you stick it in a solvent. Pretty basic. And traditionally, that gave what was called tinctures. So you take a plant material, put in some ethanol and filter off the residual plant, you'd have a tincture of cannabis or a tincture of coca or something like that. That's well and good, except it tends to leave a lot of other non-target materials in there. And if you try to pull off the solvent, you wind up with a fairly messy material that's got a lot of residual chlorophyll and a lot of residual plant cellular material. It's not really what you're after. So the general approach is you'll do your initial extraction pass. If you're from a mining background, you might refer to that step as leaching. And then you'll move into a back extraction where you're using an immiscible liquid-liquid extraction system. And the object of that is either you want to target your target product and move that into your secondary phase, or you want to target your impurities and remove them from your primary phase. Either way, it's pretty basic stuff. I'm sure you've all been across it in your courses, conceptually nice and simple, and the engineering behind it isn't too complicated. Then once you've done that and you have a relatively pure extract, your product, you either evaporate off your solvent or precipitate out a material, and you wind up with your raw material. So in the case of poppies, you call that your CPS, concentrated poppy straw, also called NRM, narcotic raw material. That could be anywhere from 70% to 99% pure. So it can be not far off an actual drug-ready material. But there's some important points around making these raw materials into an actual drug product. And I'll touch on that real briefly, but it's not the aim of the talk I want to give today. But essentially it comes down to fairly simple purification techniques. It might be a mixture of chromatography, where you're looking to use large industrial scale chromatography columns to separate out your target from the impurities, or it might be a case of precipitating out a specific crystal using a targeted conjugate base or acid and getting nice slow crystal growth to get pure crystals. A combination of those factors, there's lots of other methods that can be used to do it. But this purification step is what really takes that raw material and turns it into a drug product, an API. This tends to be done by a different manufacturer to the ones that have done the initial extraction material as well. This is where it starts becoming a lot more pharmaceutical and where regulators pay a lot more attention. So effectively, this is where the extraction industry starts interfacing with the pharmaceutical industry. So on that, what are some of the challenges involved? And there's probably 4 key points I want to touch on here. Number one, you're using solvents, and a lot of solvents aren't particularly friendly, particularly if you're going to be using chlorine-based or a benzene-based solvent. You can't afford to have any of it left in your material. And I've got a little table there, which is an extract from an ICH guideline showing that, for instance, for benzene, the residual amount that's acceptable if you've used that in a process is only 2 milligrams per kilogram. Actually getting the residual solvent down to that level can be quite a challenge. So in effect, what it means is you've got quite some limitations around which solvent you can use. And even when you pick a solvent, it can be quite hard to get your raw material within specification required to move forwards. Number 2, there's the hazards associated with using solvents. Majority of them are flammable, volatile, organic compounds. And if they're not flammable, they tend to be more toxic. So there's a bit of a trade-off there. And the net result is you wind up having much more expensive facilities to handle these liquids. And following on from that, the liquids themselves tend to be quite expensive, particularly as extraction gets more advanced and we move to more targeted specific solvents. The solvents themselves become quite expensive and recovering them becomes an important factor. So not only do you wind up with expensive, flammable, hazard-rated extraction facilities, you tend to then also have to build an expensive, flammable, hazard solvent recovery facility next door. So the capital cost for these systems is quite high, and the ongoing costs are also quite high as well. And then at the end of the day, you've got the waste to deal with as well. And the little flames I've got on the screen there are actually from an incident last month where a flammable pharmaceutical handling, a waste handling facility caught fire. And it's not uncommon, unfortunately. So not only is the process itself hazardous, but the waste will contain hazardous, potentially toxic solvents, or the waste solvent themselves will contain high schedule controlled substances. It's quite a challenge dealing with these solvent-based extraction processes. So that's kind of an overview of where a lot of industry sits at the moment in terms of botanical extraction. I want to just touch on a couple of upcoming technologies now to give you an idea of where we can potentially move and where some movements are being made. And if you're lucky, you might start working in these kinds of areas as well. So first one I want to touch on is supercritical fluid extraction. This has actually been used for decaffeinating coffee beans for about 40 years now. So it's not exactly new technology, but its use in the pharmaceutical sector is still relatively uncommon. And the primary use of it is driven through cannabis resin extraction. And to be honest, that's driven mainly from the recreational market in the US. You'll also see that modernizing economies, particularly China over the last 20 years, and some other economies that are starting to really push their more complicated advanced manufacturing, well, have started taking on supercritical extraction. The advantages there are your typical solvents, just carbon dioxide. So a spill isn't a concern. It's not a hard material to get a hold of. It's not a hard material to store. And in fact, it operates at ambient temperature under most supercritical regimes as well. So you don't need to worry too much about providing particularly heating or cooling for the system. You are operating at incredibly high pressures though, so that does become a factor of it. But even then, by modifying just a few process variables, you can actually get really specific selected properties out of this solvent. And it gives you a lot of advantages that you couldn't get even with a binary or a gradient kind of solvent system in traditional method. And it's used quite widely for plant extracts. Got a list of a few there, particularly fragile essential oils it's quite good for. So supercritical extraction is quite an interesting novel technology that's coming up, widely adopted for medicinal cannabis in the US. Starting to see a bit of adoption outside of the US in the pharmaceutical space, but still has a bit of a way to go. Next on, I want to talk on water-based extraction methods really briefly. This one's not as broadly applicable as supercritical or solvent-based extraction, but there's a surprisingly large range of ways to make water more specifically soluble for your target compound. One of them is just by simply heating it. There's a surprising change in the dielectric constant of water, which effectively determines some of its solvent abilities. By heating it up, obviously once you pass 100 degrees, you need to keep it under pressure and it starts to become a bit of an intense situation. But you can actually get equivalent dielectric constants to acetone if you heat water up far enough. So it starts behaving like a very traditional, strong organic solvent. And it is used in industry in a few spots. On the left there is a Canadian group that uses it for extracting flavor compounds out of plants and uses it for a wide variety of fruits. The bottom right, that fairly basic looking setup is actually a water-based cannabis extraction system that's used in the US. And when I was in Tasmania, I actually worked for 5 years with a joint that did water-based extraction of morphine and codeine and thebaine and oroparvin from poppies, and it was quite effective, and it can be quite a cheap process as well. You lose a lot of the concerns around solvent handling that you would have with organic solvents, but it is quite research and development intensive to figure out how to get water to work for your target compound. And the last thing I want to touch on seems a bit basic, but I think it's an important consideration, which is that you should also look to the plant you're trying to extract to try and see if there's some advantages can be had there. An example of that in cannabis is using the trichomes, which are small stalked glands that stick out on the flowers that actually carry the cannabinoids. Traditional extraction for cannabis is you take off the entire flower, you mill it up, and you try and extract that. But by some fairly simple technology, and the top center there is the Alchemist 420, a wonderfully named piece of equipment for removing trichomes. But by basically a screen box with some water and ice, you can get the trichomes off quite effectively, and you can wind up with a very basic pre-concentration step that gives you nearly a 60 or 70% pure compound before you even move to a solvent for extraction. As well as that, there's still in India traditional opium harvesting from poppies, where you lance the capsule and collect the latex. There's no industrial automated way to do that at the moment, but if there were, that could potentially be a promising avenue for an initial step into poppy and morphine and analog collection. And kind of an analogy for this is, if you're after the aspirin from the willow bark, why bother cutting down the entire willow tree and processing the whole thing when you can just strip the bark off? So sometimes it's worth taking a step back at the beginning and questioning just what our harvest procedures are and how can we make them a bit more applicable. So that's a very quick overview of the botanical extraction industry. It is a fairly major industry in Australia. It's not one you hear about a lot, but it's an important value add on top of the heavy levels of primary production we do. And for states such as Tasmania, it can be quite a big impact to the overall state's economy. And as Australia is positioning itself at the moment as effectively a leader in the medicinal cannabis industry, hopefully it will continue to be a major impact for Australia into the future as well. And the drivers for change in this industry are there. The cost of material and facility is much lower with some of these more modern technologies. The environmental impact is substantially lower, which is not only going to please any EPA applications, it's going to please legislators and it's going to please the community as well. I do have a note there that the regulators tend to be fairly agnostic to it. So your pharmaceutical regulators will be focused very much on making sure that existing processes are suitable, and that in essence can act as a bit of a handbrake on adopting some of these technologies at times. But that's more something to be aware of and something to be managed than something that should be actively slowing down any projects you're involved in. So in a nutshell, I guess that's how you make drugs. Thank you for listening. And if there's any questions, I'm sure I can take them after the next talk.
All right, thanks, Russell. We do have enough time to address one question right now.
Yeah.
So do you think there will be a shift from plant harvesting and processing to using microbes and bioreactors to produce active ingredients?
So there definitely already has been. It's very much product specific. So bioreactors are very much common for producing a lot of more modern medicines, particularly when you're talking more protein-based medicine. And like the CSL is a wonderful example of what can happen when you start moving in that direction. And if you're going to work in pharmaceuticals, particularly in the Melbourne area, there's a very good chance you'll wind up at CSL. So without a doubt, that shift is underway. But when it comes to some of these more established and widely used drug products, such as your morphines and your codeines, and potentially even THC and CBD in the near future, there is some distinct advantages to growing the plant and harvesting it directly. You can come up with some very clever processes for trying to get a microbe to simulate the synthetic processes in a natural plant. But when the natural plant grows quite readily and effectively, and you only need to cut it down and process it, it can work out being a lot more cost effective at times. So we'll see where the industry goes, but I don't think botanical extraction is going away anytime soon.
Cool, thanks for that. We also have one other question. So is it true that growing cannabis plants deplete the nutrient resources of the soil?
So it's true of almost growing any plant that you'll wind up depleting the nutrient resources of the soil. It's one of the ways in which plants grow is that they'll pull up particularly the macro and micro metal-based nutrients out of the soil. Hemp in particular grows quite fast. I can't speak to the hemp industry, but another point is that cannabis can be used as a bioaccumulator, so it can be quite an effective solution for remediating polluted sites. So without a doubt, yes, they're going to impact the soil that they're grown in. If you're growing into traditional broadacre kind of outdoor fields, then it comes back to how do you manage that land? How do you rotate your crops? How do you apply your fertilizer? How do you apply your pesticides? How do you keep that soil healthy? And that's agronomy. That's the challenge faced with agricultural countries everywhere. And what you'll see a lot with cannabis is you start moving towards indoor growth, where your growth media, a common one these days is rock wool. It's actually a spun rock that's been melted and reformed into a little block. That's what the plant's grown in, and then you just feed a liquid solution to it and it takes it up and eats it from there. So you've got very good control over what you're feeding it. So yeah, I hope that answers the question.
Yeah, it does. Thank you very much for that. So we'll now just move on to our next speaker. Alrighty, so I would now like to invite Dr. Pau Loke Show to do his presentation, and I'm very keen to hear it. Thank you.
Okay, thank you so much for having me here. All right, just a brief introductions about my university. Actually, we are from University of Nottingham. In Nottingham, we have 3 campuses. The one in UK, China, and Malaysia. All right, so a little bit about myself. Actually, currently, I'm the president of International Bioprocess Society. I'm also director of Food Processing Research Center, co-director of Future Food Malaysia, Become Excellent. Right. I also director of research from the Department of Chemical Engineering. So I got my PhD in bioprocess engineering. So a little bit background about myself. I'm not going to talk more about myself. All right. So a little bit about my research portfolio. I'm focusing on bioprocess from upstream to downstream, separation and purification technologies and engineering. And today I will talk about more to biorefining technologies. I believe in the previous few speakers talk about upstream and downstream. So I'm more focusing on biorefining, which is downstream processing. Right, this is about my research achievement. I'm not going to highlight it now. Right, so because I have a large amount of students working together with me, I have a strong research collaborations with Taiwan, New Zealand, US, Vietnam, China, Japan, and other countries as well. Right, so now to go back to my today's topic is the latest development of biorefinery in biotechnology industry. So everyone know that biotech industry is the industry that use complete or part of the organisms, example bacteria, enzyme to obtain desired products. So I believe in the previous few speaker already mentioned in the bioprocess, right? You have inoculum. Inoculum actually is a baby stage of the bacteria or microorganism. You need to take good care of it. So you have start from inoculum, then you come to bioreactors, then the bigger one. So these 3 steps is we call upstream. So after this step, right, we have harvesting tank, then continue. All this is downstream processes. Right. So you can have a look. So in bioprocess, right, divided into 3 stages. The first stage is upstream. Upstream is enzymatic reactions, cell cultivations, and plant and animals. Then we come to extractive technologies. So you can see in the middle one, we call it bioreactions. Bioreaction normally very fast, just reaction process. For reaction process, we have 3 steps. Then upstream, we have another 1 step here. So you can see in the downstream processing, right, is very long process. Is is complicated process. So our idea is to minimize the downstream processes. So in biorefinery, what is biorefinery, by the way? Biorefinery is a facility that integrate biomass and the other equipments to produce a fuel, power, and value-added chemical from biomass. So in biorefinery, we divide into 4 stages. Okay, the first stages is capture of the inoculum, which is you capture the insoluble microorganism, insoluble, whatever that insoluble in solvent in water, you capture it. Second one is a product isolation. So you remove your product into another stages, so we call it product isolation. Then we come to product purification. You purify our products. Then last stage is polishing and product packaging. So it's important or not, downstream processes. Yes, it's very important because it's equivalent to refinery in petrochemical industry. As you know, in Malaysia we have a huge petrol industry, which is oil and gas industry. So this is considered very important process. So same thing to in the bioprocessing industry. Right, in conventional downstream processes, right, we divide, you can see we have a chromatography, liquid extraction, precipitation, distillation, membrane technology, and electroanalysis. So all this, right, you can see is we call it conventional technologies because we using the previous people to already well developed technology. Now we're going to improve it with the new technologies. Okay, so in biorefinery principle, we divide into 4. The first one is based on the size. Okay, you can see the small size can do the separation effectively, can go in, while the middle size, maybe some can, some cannot, while the big size of course cannot. So the factors that affecting refinery principle is size. The first thing is size. Second one is based on the hydrophilic, okay, light water on this light water. Okay, so you can see some light water you will attach to it, some this light water you will bound to each other. So this I can see this another principle to do biorefinery. The third one is the negative positive charge or cation or anion. Okay, you can see. Okay, the negative charge normally you can bound to each other, positive and negative you will attach to each other. So this is another principle they're working in biorefinery. So using this process in bioseparations. So the last but not least is something like enzyme, key and log, or antigen and antibody. So depend on specific affinity of the binding. Alright, so you can see only the specific binding can attach to the surface. If not specific, right, you cannot attach to the surface. So in total, we have size, we have hydrophobic, positive, negative charge, and the surface antigen and also the antibody size. Okay, so this is the 4 biorefinery principle. All right, so today I would like to introduce a new biorefinery method. We call it liquid biphasing system. Actually, the previous speaker already mentioned that you have supercritical separations method. Then you have an aqueous phase method, separation method. While this new method is considered liquid biphasing system, is considered aqueous biphasing system. It's a new separation method. All right, so this liquid partitioning method is a liquid partitioning method of 2 liquid phases. Why I say so? Because you only mixing of 2 liquid phases. So this method really successfully recover and separate many bioactive compounds. For example, color pigment, protein, carbohydrate, lipid, vitamin, mineral, enzyme, nutrient, aroma, heavy metal, and nanotube. So you can see whatever that you name it in this in bioactive, right, you can separate it. Also, you can see even not bioactive compound such as heavy metal nanotube is not bioactive compound. You also can use liquid biphase system to do the separations. Alright, so liquids, in order to prepare liquid biphase system is very simple. Just need 2 steps. Just how you make coffee. Alright, so if you make coffee, you need creamer, sugar, milk, whatever things, right? You just put inside your coffee maker, you mixing it, then you set it under gravity force, you will form 2 phases. So in this liquid biphase system, the phase forming component is very important. Why I say so? Because the phase forming component can be cell destruction agent, will disrupt the cell, and also can be separation and purification agents. Right? So another factor that affecting liquid biphase system is concentration of the phase forming component. If below the critical concentration become miscible homogeneous single phase, you're not able to form 2 phases, just like 1 phases here. Okay, if above the critical concentration, above the phase here, then you form 2 phases. If you become turbid and form 2 phases, this is an example of this PEG, polyethylene glycol. So weight per weight. So the concentration of the PEG and the concentration of the DEX dextran. So you can see the concentration of it, right, play important role. So whenever you prepare the liquid biofreezing system, it must above the curve. If wherever you prepare below the curve, then it's not considered two phases. All right. So why is so important? Why is so powerful of this liquid biofreezing system? Right. So everyone know that in bioseparation, right, it divides into fermentation plus cell harvesting. Okay, so upstream and downstream. Upstream is the fermentation plus the cell harvesting, while downstream is considered of cell destruction, purification, and polishing. So it consists 80% of the total manufacturing cost. So the advantages of liquid biofreezing system is high water content, because this only consists of 80% of the water content. It's not like oil and water. Oil and water consists of less water content, and also low interface tension. Because like I mentioned, oil and water high interface tension, so that the biomolecular difficult to migrate from top phase to bottom phase to bottom phase to top phase. So the low interface tension will easy for the microorganism to migrate from top and bottom to top. So also rapid processing, less than 10 minutes. I don't think you make a coffee more than 10 minutes, right? Definitely very fast processing. Stabilizing effect, special property of the phase forming component. Also large scale application, you just mixing of two liquid phases, then you can scale easily. Simple process, just shaking and centrifugation. Low investment cost, because all the chemical they use in liquid biofreezing system can be recycled. All right. The method that use the chemical that we use is environmental friendly, because we deal with food industry. So in non-toxic forming component, and of course it's already proven high yield, high recovery, high purity. This is very new method. I can say it's the latest development. So we in this way right, liquid biofreezing also have an improved version. We call it thermal separating biofreezing system. Also integrate with bubble, we call liquid biofreezing flotation. Also integrated with upstream and downstream, we call extractive technology, as I mentioned. Because you combine upstream and downstream together, then you can save a lot of time, cost, and make the process more simple. Alright, how thermal separating biofreezing system work? Actually, it's very straightforward. Okay, you can see we put one phase mixing of your ingredient like fermentation brew or microorganism cultivation, plus the solvent that you want to put, like for example alcohol, polymer. Then we heat it above critical solution temperature. In that case, you cannot heat too high, because we are dealing with live organism. You will denature if it's too high. So we combine this. So after heating it, right, you will form two phases. The top phase consists of water rich top phase, while bottom phase consists of EOPO rich top bottom phase. So the EOPO rich top bottom phase can be recycled, and while the top phase, you just remove the water, you can get your pure product. Right. The second one I want to introduce is the liquid biofreezing flotation system. Liquid biofreezing flotation is actually combination of the idea of solvent sublimation plus liquid biofreezing system, come up with liquid biofreezing flotation. Alright, so you can see solvent submission is a bubble concept. Liquid biofreezing system is a concept that I mentioned just now, using the thermal separating concept. We heat it above the critical solution temperature, we form two phases. So to combine solvent submission and liquid biofreezing system, we come up with liquid biofreezing flotation. The gas passing through a cinder glass disc, we form a bubble. Because of hydrophobic hydrophilic, all the active compound will attach to the surface of bubble. The bubble will flowing to the top phase, which has solvent to extract the active compound. So you can see this is the first layer of separation, then come to the phase forming component, second layer of separation. So you have two layers of separation happen at the same time, so the purity can be reached up to 99%. So there is a lot of factors that affecting liquid biofreezing system. Just like how you make coffee, you need to have the correct ratio of coffee, creamer, sugar, the temperature of water. So the same thing to liquid biofreezing flotation system. You need the molecule of the protein, polymer, concentration of the salt, concentration of the polymer, pH, volume of the aqueous phase, volume of the polymer phase, amount of the crude feedstock, amount of biomolecular biomass you put inside, gas flow rate, flotation time. The more parameter you optimize, the more pure product you will get. So you can see. So we're passing a cinder glass disc, we form bubble. Bubble we have this is a carbon dioxide bubble. We attract at the surface of bubble. This EOPO, which is ethylene oxide propylene oxide, is a type of polymer. We remove it, heat it up to critical solution temperature. We form two phases. The top phase is a pure product. Alright, it's a lipid Then the bottom phase is concentrated EOPO, which you can recycle. Alright, so let's continue with extractive technology. Extractive technology actually is something like one stone killing two birds. Alright, so you combine upstream and downstream together. So how it works is just first stage, you just normal cultivation, but you add phase forming component plus the feed butter. Like example, the biomass I will grow is microalgae. So we put everything inside a one strict flux. We grow it together. Alright, then after the harvesting, then maybe 1 week, harvesting time. So you stop the agitation, you heat it above the critical solution temperature, 40 to 50 degrees Celsius. Then you will form two phases. The top phase is bioproducts, which is product you want, plus the water. You just use the distillation to remove the water, then you get your pure product. While bottom phase is the concentrated phase forming component plus the biomass, which you can recycle and reuse it for the next cycle. So this is we call extractive technology. Alright, so we also integrate with algal cultivation. You can see this is the technique that we use in our lab here. So you can see we use cultivation using bubble concept. So as you know, in microalgae, we have a great potential. It can produce a lot of biofuel, supplement, pure protein, DHA. All this you can produce using this algal cultivation, using this technology. And also, this is a few of the journal that I would like to share with you. I'm the editor for these few journals. Right, I think this is all from my slide. Thank you so much.
Thank you.
All right. Do you have any questions for me?
I actually have one question. At the end there, you have the bioproducts are in the water with the biomass phase, the two different phases, and you mentioned that to separate the bioproducts and the water, it's through distillation.
Yes.
What happened? Can you use the same kind of process, the distillation process, if the bioproducts are actually heat sensitive, or would you not use this approach for extracting those kind of products?
All right. So for your information, right, you can see this is a previous slide. All right. All right, so you can see this is the previous slide that I mentioned, right? In the top phase, right? Top phase, normally, if you use alcohol, you can use distillation to remove and to remove the solvent, then you can get your pure product already. Actually, there is few ways to do it. Depends what is the top phase that you want to use. Let's say you want to use polymer, then you don't need distillation column. While if you use alcohol, then you have to use distillation column. So it depends what top phase that you want to use. Yeah, am I answer your question?
Awesome. Now to wrap up the talks on pharmaceuticals and biotechnologies, introducing Dr. Charles Cooney, Professor Emeritus and Co-Director of the Program on the Pharmaceutical Industry at the Massachusetts Institute of Technology. Professor Cooney also sits on the boards of Genzyme, Polypore International, and Biocon Limited, and has published over 250 research papers over his storied career. Professor Cooney, thank you for joining us. I'll hand the reins over to you to talk about biotherapeutics manufacturing to meet patient needs in 2030. Take it away.
Thank you very much for the introduction. I'm delighted to have the opportunity this afternoon to speak with you, while unfortunately not in person. Electronically is working very well in these stressful days of our pandemic. But most importantly, I'd like to share with you my excitement about not just where we have been, but where we're going. I've had the privilege of being involved with the biotech industry since the discovery of recombinant DNA technology and the first companies to emerge in the early 1980s. And I must say, as a chemical engineer, as an academic, to be involved in the building of a new industry is unbelievably exciting as part of one's career. But where we are today in 2020, we are at one of the most exciting times in biotechnology and building of the biopharmaceutical industry. And it's that challenge, it's that excitement that I'd like to share with you. And the challenge is really yours. How are you as engineers going to build the biopharmaceutical industry to meet the needs of the patients, not just today, but in 2030, well into the future. And as we look around us during these pandemic times, you can see the importance of this, the criticality of your role as we go forward. When we look at the industry today, we see it's under pressure from all directions. Patents on the blockbuster drugs that generated the profit to fuel the industry, they're expiring. The productivity to develop new drugs for unmet medical needs has been low. The pressure from costs, the pressure from the need to provide access to healthcare globally, and of course, most recently, the COVID-19 pandemic is putting a tremendous pressure on our ability to deliver to meet those healthcare needs. And of course, the change into the industry itself, how we prescribe, the opportunity for precision medicine, and the opportunity to cure a disease, not just treat it. These are the conditions that we operate in today. Now, let me draw upon one of my hobbies, which happens to be high altitude mountaineering. And there's a lesson that I would like to convey from this. As we embark on a journey, which all of you are, there's uncertainty and there's risk. And we need to distinguish what are our aspirations as we embark on driving the new technology for the benefit of society and the patient going forward. What are our aspirations? And it's thinking about those aspirations that was on my mind when I took this picture. At this point, I was an altitude of about almost 6,000 meters, and I wanted to ascend Ama Blam, a mountain in the Khumbu region, just south of Everest. And as you look at this journey, you see that it is fraught with risk. Those rocks, that ice, the sheer high altitude. But it's not the risk that concerns us. The lesson that's important to learn is, what is your goal? So as I stand here, working my way towards the summit of Ama Blam, what am I thinking about as I look at my feet? Well, I'm thinking about my next step, because if I take the wrong step, the downside is not so pretty. I'm thinking about the summit, which I aspire. But those are not my goals. My goal is to get back down. Keeping in mind that my climbing partner is my son, and if he has a problem, what am I going to say? Oh, just sit here, wait for me to come back. No, my goal is to get back down, even though I have an aspiration to summit. So as we think about the biopharmaceutical industry, what is your goal? And I would offer that the goal is the sustained delivery of a safe, affordable, efficacious medicine to the patient. Safe, affordable, and efficacious medicine to the patient. I would further argue that we want to provide this to all patients in all regions of the world. But as an engineer, as we think about this goal, we think about manufacturing. And manufacturing lies on the critical path between the exciting science that's being developed in the lab and the patient. If you can't manufacture it, the science doesn't do you any good. So let's keep this in mind. And throughout my career, and my career spans 50 years, I have focused on manufacturing. How do we take the science of biology and translate that to the benefit of the patient? That's manufacturing. So I offer a framework to begin thinking about what you should be doing. And that framework begins with why, what, and how. Why do you want to change what we're doing today? It's not working so badly. What is it that's happening in the lab that will enable us to deliver new science to the patient. What is the vision for advanced manufacturing, the factory of the future, to meet the needs of the global population? And importantly, how will you implement this change? And to do that, the framework that I offer for engineers is to think about the science through 3 lenses simultaneously. That's hard, given that we only have 2 eyes. But you need to think about how it gets to the patient and the business that will underlie the sustainability of that vision. And your job is to manage the resistance to that change. Now, this is not a new challenge. When we look at the chemical industry over the last century or more, we see that for multiple industries, steel, chemicals, food, and yes, pharmaceuticals, these industries are driven by a business need. They need to reduce cost. They need to enhance speed to market. They need to assure quality. And they must have flexibility because demand is variable. So we do this by moving from traditional batch processes to continuous or integrated processes. Can you do this for the biopharmaceutical industry? Now, as I have reflected on the industry, there are a couple of lessons, 2 in particular, that I would like to share with you. And as an engineer, we think about technology, we think about science. This is what we're trained to do. But lesson number 1, it's not about the technology, it's about the business case. And when we think about what we want to do in building new manufacturing capabilities from new science, or improving old capabilities from existing processes, there are 4 metrics that I find very helpful thinking about how well we're doing. And those 4 metrics are speed, cost, quality, and flexibility. So when we think about the business case, which is really driving the value proposition, and for us, it's the needs of the patient. That's what we seek to do. That's our goal. We need to think about speed. Speed is how fast do we get a new therapeutic to the clinic? How fast can we transfer the technology into manufacturing? How fast can we develop a process for productivity? How fast can we scale it up? And this we see this dramatically today in the case of vaccine development for SARS-CoV-2 to deal with COVID-19. How do we assure quality, the safety, and the efficacy to the patient? How do we manage the cost? How do we manage the flexibility for uncertain markets and our ability to meet those markets in multiple cultures, multiple business environments, multiple countries? And the challenge that I would put literally in your hands is, how do we do this for a disease such as COVID-19? And how do we do this for any unmet medical need? That's the first lesson. We need to understand the business case. The second lesson is taken from the example I mentioned a moment ago, and that is, how did the steel, the chemical, the food industry evolve over the last century or 2? It evolved by moving from batch processing to continuous processing. So isn't that what we want to do today? Well, I took an excursion about 2 years ago in preparing for a paper that I was to deliver. And I called up my friends, colleagues, connections within the industry, and I asked them this question. How are you going to move your traditional batch processes to improve speed, quality, cost, and flexibility into continuous processes? And I learned a lesson. And the lesson was I was asking the wrong question. It's not about continuous, but the challenge we face to meet the demands of the future are about intensification of our process, our technology, integration of the individual unit processes that constitute our overall process, and interrogation. How do we measure and control in order to meet the delivery of the quality drug product to the patient? So the how of the why, what, and how is not about continuous per se, but continuous is simply one mode by which we can do it. The how is implementing process intensification, integration of the process, and interrogation to meet the business need. So this is an important lesson that I offer you as you think about your own careers and the development. And let me build upon this. What are the attributes of intensification? What do I mean? Well, it can be thought of in the following way. We want to increase the capacity across our each unit operation in our process. We want to increase the capacity of each individual step in our process. That's intensification. We want to utilize that capacity by achieving high productivity. We can do this by improving the biology of our process, by improving the chemistry of our process, by improving the physics of our process. We need to reduce the time it takes to not only reach the clinic, but to build that process so that it has a scale suitable for the population that we wish to make. A good example, again, is COVID-19. Is it good enough to make a vaccine that will address 100 million people? No. 500 million people? No. 1 billion people? No. We have a world of 6 billion. And we do this as engineers by thinking about fundamental things like our reactor, by increasing the reaction kinetics, and I'll come back to that. We are able to reduce the residence time in our reactor. That's the speed benefit. And when we have both speed and productivity, this allows us flexibility. So these are the components one thinks about in process intensification, whether it's your reactor or whether it's your individual steps for purification in producing the product. What are the attributes of process integration? We want to simplify our process. Simple is good. In the case of a bioreactor, which is the heart of where we're making a biological product, we want to increase the concentration of our catalyst, the actively growing cell, and that's going to increase our productivity. Turns out that's not so easy. It's a problem in biology. It's a problem in chemistry, how we maintain the right environment. And it's a problem in the physics. How do we supply the oxygen? How do we remove the heat? How do we assure an environment that is conducive to the growth of the cells? And we do this by techniques that allow us to retain cells in the bioreactor. Fundamental to process integration is reducing the interfaces between steps. Problems happen at interfaces when you go from a reactor to a separator, or a cell separator to a chromatographic step, and so on. Process integration is intended to reduce the size of the plant, to reduce the footprint, simply to reduce our capital cost and our operating cost, our CapEx and our OpEx. And one of the things that has become a hallmark of modern manufacturing is modular design. So we have advances in how we design, build, and implement facilities that we need to bring together to implement process integration. Well, let me speak to a particular example. This is a typical process, batch process, for production of monoclonal antibodies. And if we go from a batch process shown by all these individual steps to a continuous process or an integrated process, what you see as an X represents everything that's removed, greatly simplifying what we do. Everything else that's part of the process becomes smaller because we don't have to hold things for a long residence time. The result is a simpler process, a smaller process with lower cost, and very interestingly, higher quality, because we reduce the residence time for our reaction in the reactor. The opportunity to make side products or create variants in our product is reduced, and the quality of the product that is ultimately produced is improved. And because we're operating at a smaller scale, we have much more flexibility as well as reduced capital cost. And this gives us much greater opportunities to scale the process, transfer the technology across many parts of the world. So these are the things that we can do as we think about process integration. Well, what about interrogation? What does that mean? Well, a product is defined by its critical quality attributes. These are the chemical and the physical and the biological properties of our product that are critical to their delivery of value, therapy to the patient. Do we know what they are? Can we measure them? Can we control them? This is the challenge to the chemical engineer. And once we begin to be able to measure, this is the precursor to learning and how we improve the process. And with learning, we can begin to build models that allow us to do computational experiments, which are much less expensive than physical experiments. And once we begin to learn and we have models, we can implement artificial intelligence. We can begin to implement machine learning. We can construct a digital twin that allows us to improve the process as we go forward. So this is where chemical engineering in terms of process integration is today and where it's going to be continuing in the future. So let me go back and briefly talk about the business case, because the business case is going to drive the research, the development, the manufacturing that you do. If it doesn't make business sense, then there's no way to support and sustain the industry. So I'm not going to speak to all the individual components in this list, but this is a shopping list. Depending upon who you are, the nature of your business, the country that you're in, who pays reimbursement for the drug products that you're making, what is the state of your technology, how are you trying to compete with your competitors? Those answer to those questions will drive whether your business case is about improving your product pipeline, whether it's about pressure coming from the marketplace to be more cost competitive, or whether it's simply the business case to build a test facility and de-risk your capabilities. But you need to understand as an engineer that business case, because that's going to drive your technology forward. And let's go back to intensification and integration and realize that in any technology, typically it's not one size fits all. We have a complicated process of multiple unit operations. And it may be that your business case tells you, I just simply need to make the upstream part of my process continuous because my downstream works fine. Or perhaps the challenge is reducing the cost of this very expensive set of steps intermediate in my process. Perhaps my business case is connecting my synthesis to the initial recovery. Perhaps it's end-to-end continuous for the entire process. So as engineers, we have a multiplicity of strategies and tools that we can bring to bear. Well, let me take you quickly through two cases. And these are examples of cases that come from conversations I had with individual companies. Thus, I can't speak to the details of the product, but these are real solutions that these companies were putting in place. So the first case is that they had a very successful product, and they were having difficulty with their existing process to meet the demand for that product. So this is the upstream process, the USP. How did they solve that? And this particular product had a unique feature that it was unstable, that as you carried out the biochemical synthesis in the reactor, if you left it in the reactor too long, it was degraded. And you can imagine what that did to the demand for the product. So their business case was that the downstream process was working perfectly fine, but they needed more product and to prevent its degradation. So they designed a cell retention system for the reactor. It's called perfusion technology. They were able to achieve very high cellular densities, more than 100 million cells per milliliter in the reactor. And as a consequence, they were able to reduce their reactor size from a batch process to a continuous process by about 50-fold. And you can imagine what that does to cost. So process intensification enabled them to meet the demand and reduce the cost. A second example is just the opposite. The company's upstream process, they had improved it phenomenally. They were making more product than they could recover. So the upstream was fine, but the business case demanded that they have a downstream process that could handle this high productivity upstream. And the management challenge was to increase capacity, but not to increase capital cost. What they did was to integrate the downstream process with continuous chromatography, viral inactivation, which is an important safety step, and membrane filtration using a rather new technology called single-pass filtration. In addition, they were able to implement very effective process control. And when they did this, their downstream process was able to meet the demands of the market because they could now recover and purify safe and efficacious product for the patient. So I want to close with two more slides. And this is what I call the call to action. This is your job. This is what you as engineers need to do to meet the needs of the patient in 2030. This is your career. You need to have clarity in the value proposition. What is the value? What is the impact of what you're doing? You need to think about, how do I intensify? How do I integrate my process? Create capacity, create flexibility, reduce cost, increase the speed to market, and of course, assure robust quality. You need to think about how to apply a digital strategy of measuring the right parameters, controlling them in the right way, learning about the process, applying machine learning, and truly make a chemical process not just better chemically, biologically, and physically, but operationally. And that's the essence of interrogation and the digital strategy. And mind the gap. Avoid interfaces. Things happen when you move from one step to another. Things happen when you have to bring disciplinary principles from chemistry, biology, and physics together. And if you're not capable of operating at those interfaces between the chemistry, the biology, and the physics, then there's a gap. And your success is not assured. So this is the challenge to you as individuals. So in closing, let me go back to my analogy with the mountains. My son and I were successful in summiting Ama de Blanc, 7,000 meters. And it was a beautiful day, and you can see a view of Mount Everest here in the background, only 10 miles away. And there we were on the top, feeling quite good. But the lesson is, when you're on the top of the mountain, you're only 25% into your journey. 75% of the accidents happen on the way down. When you have developed a manufacturing process, when you have built a bioreactor, when you have built your pilot plant, when you have built your plant, you're 75% into the journey. Because unless you can deliver that product to the patient successfully at a cost that people can afford, at a quality that assures safety and efficacy to the patient, with the flexibility to meet the diverse demands of a global community, and with the speed that is necessary, that is driven by disease, then you're only, unless you can do that, unless you can do it successfully, you haven't met your goal. And that's a challenge that you have as engineers. And I share that excitement. I have been part of this journey for decades. And I am delighted to pass the baton to your capable hands to deliver that goal, to meet the patient's needs in the future. Thank you all very much. And my best wishes for your health, your safety, and your success in your journey.
Well, thank you, Professor, for your absolutely fantastic presentation. I know myself as a pharmaceutical sciences double degree student, and I know so many people watching this have some very valuable lessons to learn from you. Thank you very much for being here. Thank you very much for taking your time to do this presentation for us.
Best wishes to all.